In this video, I'll be going over Hershey and Chase's revolutionary bacteriophage experiment. But first, a brief overview. In 1952, Alfred Hershey and Martha Chase published a paper indicating that DNA was the genetic material for bacteriophages. They used T2 bacteriophage that infects E. coli. The phage reproduces by attaching to the outer wall of a bacterial cell and injecting its DNA into the cell. The DNA is then replicated and directs the cell to synthesize phage proteins, producing more phages that break open, i.e., lyse, the cell. The phage reproduction process was unknown at the time of the experiment. It was known that bacteriophage is around 50% protein and 50% DNA, and that phage infects a cell by attaching to the cell wall, then producing progeny inside the cell. Since progeny carry the same traits as the infecting phage, the genetic material from the infecting phage must then be transmitted to the progeny. The how of this was unknown. The purpose of this experiment was to determine whether DNA or protein is the genetic material carrier. They wanted to see if protein or DNA would be transmitted into the progeny phage after lysis. The experiment. Hershey and Chase designed a series of experiments with isotopically labeled DNA and protein to track which is transmitted in phage reproduction. Radioactive isotopes can be used to identify the location of a specific molecule, as anything containing the isotope will be radioactive and easily detected. DNA contains phosphorus but not sulfur, so Hershey and Chase used radioactive isotope 32P to label phage DNA. Protein has sulfur but not phosphorus, so the phage proteins were labeled with 35S. Two E. coli populations were grown. One batch was grown in a medium containing 32P and then infected with T2 phage. All progeny phage then had isotopically labeled DNA. The same was done with the second group, but 32P was used, so progeny with isotopically labeled proteins was then produced. The researchers then infected two unlabeled batches of E. coli with the 35S and 32P progeny phages. After allowing for the phages to infect the bacteria, the cells were placed in a blender which removed the empty phage proteins from the cell walls. The protein coats were then completely separated from the cells using a centrifuge, and the infected E. coli were cultured. Key findings of this experiment. Hershey and Chase found that for bacteria labeled with 35S, nearly all of the radioactivity was detected in the protein coat, and when the new phages emerged from the infected cells, they contained virtually no isotopic label. In contrast, the E. coli infected with the 32P labeled DNA were radioactive, and when the cells were lysed, most of the progeny phage was also radioactive. This demonstrated that DNA from the infecting phages had been passed on to the progeny phages. The results of this experiment had the major impact of confirming that DNA, and not protein, is the genetic material in bacteriophages. This experiment then paved the way for further research into DNA and its role as genetic material.